Okay, once again, I just spent the last few minutes talking to nothing because I was not recording on this computer. Let's just make sure, pause, stop recording. Yes, we are recording. Okay, so it's Sunday. I wanted to get to this on Saturday, but I just didn't have the heart to it. But the good news is I got more done with my the shed that I'm building, which I'm sure you all care about. So today, look, guys, this is brain. This is hard stuff, and I kind of feel like we went from liver, kidneys, where the anatomy is kind of there, and like, okay, yeah, I can get it. Suddenly, brain, and it's rough. Suffice it to say, we have next semester where we really look at OBGYN. We are going to be really looking at the brain a little more closely next semester. So if you're still feeling a little rough around the edges on this, the next semester will help. So keep that in mind. Having said that, some of the anatomy that we worry about, let's say like cavum, septum, pellucidum, is more of a OBGYN concern than it is a neonate concern. So there is a lot of crossover there. What else did I want to say? All right, well, I'll think about it when we get there. I've got a few extra images and things that I wanted to get a little more into, or I thought I was rereading the chapter, only the first part of the chapter, guys. We'll catch up on this stuff quickly. Don't worry about it, but only really the first part of the chapter, and because the anatomy is the hard part. Once you get basic anatomy, and then intracranial hemorrhage, which is I'm seeing is the very first thing that we talk about, which is the most important thing we do. Hemorrhage, hydrocephalus, and that kind of stuff is easy once you understand the anatomy a little bit. So that's what we'll do. I just want to kind of go over. Well, I'm going to go over the same exact page. I'm going to go over it a little faster this time. And like I said, there's just a couple more images that I add to it. So without further ado, let's get that going. Okay, back to brains. Yeah, I'm just going to work my way down. Once again, guys, let's do a new color. For anything I write today, it's going to be a different color. Okay. And we are on the wrong pen. Okay, now we're on the correct pen. All right. Cerebellum is basically your brain. The whole brain is the cerebellum. It is in two halves, separated by a interhemispheric fissure. There is well, let's do this. Let's go continue to talk about a couple of different lobes. We have the cerebellum, which is kind of almost you can see it's a different part of the brain entirely. It's shaped differently. It's built differently. It is kind of a primitive part of the brain, as well as the brainstem is primitive. The cerebellum back here helps coordinate our movements. This lobe, and it kind of goes off to the side, is the temporal lobe. We have back here the occipital lobe, part of the cerebellum, as well as the temporal lobe. Part of the cerebellum is kind of the overall structure. And then we have different lobes: temporal lobe, occipital lobe, parietal lobe. Let's just front. That says frontal lobe. It looks like I need to unlazy myself and sit up straight here so I can draw and write straight. And then we have a brain stem, which extends further into the brain than we see in this picture. That is, and then connects with down all the way down here into the spinal cord. The brain has sulci, which are the grooves or the valleys, and gyri, which are the ridges. They're basically one structure of a ridge and a valley. This increases surface area. This is where all the thinking occurs. Thinking right here in the sulci and the gyri. You don't think in the middle of your brain. You think on the outside of your brain in general. Let's see. Corpus callosum. That was a really important part of the brain. That is this structure. Horrible, can't quite. All of this structure here. That is the bridge between the two hemispheres of the brain. I might have said something incorrectly. In fact, as I remember it, I think I did say something incorrectly. So this is a oops moment. I believe that I said that this structure here, yeah, in fact, I remember now, I did say that this structure here was, you know, kind of like we're cutting into the lateral ventricle. And I don't believe that is correct. I believe that this structure, let's get rid of, well, no, we can go ahead and leave that. We'll go ahead and leave all that blue there. That is the cavum septum pellucidum, CSP, cavum septum pellucidum. Now, if it goes further, actually, I got a picture. I got a picture of this. If it goes further back, that's more of a primitive. That's more of a premature babies might have an extension of the cavum septum pellucidum that goes farther back, and then that's the if that exists, then that would be the cavum vergae. And that's, let's see, only very premature. Okay, now one thing I did say correctly, and I double checked, cavum septum pellucidum, and for that matter, the cavum vergae, are not actually a part of the ventricular system. They are potential spaces. Now, potential meaning everyone has it. Every baby, every fetus has a cavum septum pellucidum and a cavum vergae. The cavum vergae obliterates first, and so we usually don't see that in the neonate. But the cavum septum, and sometimes we don't see it at all in the fetus. That's a kind of an optional. It's an optional space. Cavum septum pellucidum, pretty much we all have that early on in development. It is actually filled with cerebral spinal fluid, but it's more of a incidental, like it's leaked into that area. So the cavum septum pellucidum is its own space. Even though it is filled with cerebral spinal fluid, it is not considered a part of the ventricular system. And so, and by the time you are a a few years old, you probably don't even have a cavum septum pellucidum anymore. It squeezes shut and is no longer there. And anyone in this class, very unlikely to have it. If you actually do have it, that could explain some things. No, I'm just kidding. But that the persistence of the cavum septum pellucidum into adulthood is associated with some problems. Let's see. Oh, the cerebellum. I do have a new picture for the cerebellum. So, and I also have a new picture for how the fluid, cerebrospinal fluid, gets into this into the cisterna below the cerebellum into the cisterna magna here. So we'll go over those two images. So the cerebellum is kind of its own. This is its own little different looking piece of brain here. And I've got a new little picture here to show you. And here's the cerebellum. Once again, looks different because it is different. It's a very primitive part of the brain. And this red area here is the vermis or veriform veriform cerebrum or cerebellum, but the vermis is, that means worm-like. And so anatomists, when they really look at this closely, it kind of looks like a worm. This guy doesn't always show up to the party. If you don't have a vermis, you have something called Dandy Walker, which is an aberration of the brain, something you don't necessarily want. But so agenesis of the vermis leads to some issues. Now let's see. Oh, do I want to go over that right? Let's talk about, yes, let's do this right now. It's the same picture. I've already have all sorts of horrible drawings on it. I'm sorry about that. But, and remember, I showed you the other day, there is a three-dimensional spinning lateral ventricles. There's two ventricles. You're basically seeing one here, and then the other one is shadowed back here, but that's just showing trying to show a three-dimensional image. These ventricles are, when you're dead center, when you are dead center on the brain in the sagittal, you should not see the ventricles. You should only see the third ventricle. But the moment you sweep your transducer to one side or the other, then you run into these guys. So in ultrasound, you probably aren't going to see all of this at the same time because these are a three-dimensional object. You are, as an ultrasound tech, you are looking at a two-dimensional slice of anatomy. So dead center, you see the third ventricle. And by the way, you don't see it like this at all. It is a slit. It is a thin slit. And if it's not a thin slit, then that's evidence of some other kinds of issues where the third ventricle is too big. So you don't see the eyeball and this little weird beak and the neck and all this funny stuff that looks like a crazy alien bird. But when you go off to the side, you will see the lateral ventricles. And of course, we have the frontal horn or anterior horn, same through the frontal, or I'll just put ant for anterior, frontal or anterior horn, and body. I mean, no one uses the term body. I'm just going to scratch that one out. And then it comes back to all the way down to the occipital horn, which is all the way back in here, that guy there. And temporal horn, this is a little more off to the side, by the way, so you can't really see this on this two-dimensional image, but this is out towards us a little bit more. That's the temporal horn. And then this area here where the three sections meet is called the trigone. That's the trigone here. They connect through the foramen of Monroe, the frontal horn through the foramen of Monroe, which you will not see on ultrasound. Just know it's there, to the third ventricle, which you will see as a tiny little slit. I think I have an image that I can show you of that. And then there's this aqueduct. This aqueduct is so thin that on a normal baby, without without an increase in size of the ventricular system, you will not see it because it is too thin. It's called the aqueduct of Sylvis, and that's from basically here down to here. You will see, if you're good, you will see the fourth ventricle. Oh, and I looked up, okay, so we have the third and the fourth ventricle. Well, what the hell are the first and second ventricles? It's the lateral ventricles. And so if you read from right to left, you say the right is first and left is second, then that would be the right ventricle would be the first and left ventricle would be the second, or vice versa. It does not matter. What's creating the cerebral spinal fluid? The choroid plexus. And we do see these choroid plexus as echogenic structures. Like I was saying the other day, this is really drawn outside of the ventricle. And I was a little taken back by that. And I think I said, gee whiz, when you're doing an ultrasound, it really seems like it's on the inside. Well, I was just doing a little bit of research. And the article I was just reading by people smarter than me were referring to the choroid plexus being within the ventricle. So, take this picture as it is, take the ultrasound picture as it is. It certainly appears to be inside the ventricle, and I do believe it would be inside. Because if it's producing the cerebral spinal fluid and basically leaking cerebral spinal fluid outside of it, it's going to leak into the ventricle. It kind of needs to be in the ventricle. So that makes sense to me. Now, this ventricular system is connected to the outside of the brain. And it's also connected to the spinal cord. You see that down here. But it's also connected to this space back here. Oh, wow, that's what happens when I use my mouse to try to draw. Oh, sorry, nose itch again. So I did grab another picture. Where did I put it? Oh, over here. Over here. There's a couple of pictures I want to talk about over here. Look, the only reason I bring this up is because the book mentions the foramen of Luschka and the foramen of Magendie, and the book just throws it out there as if you're going to know what it is, and it kind of bothers me when they do that. But these foramens, whatever, are allowing cerebral spinal fluid to come from the ventricular system into other parts of the system. So the foramen of Magendie, this is going to bring our fluid into a reservoir. It's not a ventricle, but it is a reservoir of fluid called the cisterna magna. So that's how fluid gets back to the cisterna magna, and then that connects to the whole outside of the brain. Now, speaking of outside the brain, and so I mentioned these granulations, and I don't think I was very clear. So let's make this a little more clear. I talked about granulations. I hope I said arachnoid granulations, but that's what they are. Arachnoid is in Spider. Yep, Spider-Man. because microscopically they look like spider webs. I guess that looks just like a spider web, doesn't it? The arachnoid granulation is where cerebral spinal fluid, and in this case, called a subarachnoid cavity, but the cerebral spinal fluid is green here. They pop through, and in this case, the superior sinus, which by the way looks a little more like this. It is kind of almost a triangular vein. It's a vein that, of course, is shaped to the surface, or is shaped to the cavity it's in. This superior sinus travels, I don't know if you guys can see me, from kind of all the way through the center of the skull. And so this is the falx cerebrae. In other words, this is the interhemispheric fissure. This is, let's just say, from an imaging perspective, this is the right hemisphere. This is the left hemisphere. This is the fissure between the two hemispheres, and there's a large vein that runs through the middle of this. When I was brand new to imaging of the brain, I brought this to the attention of the radiologist. He says, "Very good, you found a completely normal structure. That's the sagittal sinus." I was a little embarrassed, but I didn't get a lot of training on the brain. So anyway, yeah, and so this green cerebral spinal fluid, it needs to be returned to the venous system, just like your lymph fluid needs to be returned to the venous system. In the case of lymph, all of your lymph throughout your entire body, it all eventually drains to your two subclavian veins, and there's literally just dumps in, and that becomes part of the blood again. Well, cerebral spinal fluid does the same thing, except it does it in these arachnoid granulations, and here is one that is actually going into the superior sagittal sinus. There are others, and this is where I kind of freaked out. There are others that do this. Another granulation, just like this granulation, except this granulation is over here. Well, as you could see, I indented the skull here. Well, when you look at an X-ray, probably more of a CT of the skull, and you look carefully, there's all these little holes on the skull, on the inner skull surface. And I asked a radiologist once, what the hell am I looking at? And he says, oh, those are arachnoid granulations. And I was like, oh yeah, sure. And I pretended like I knew what he was talking about, and then I went and looked it up and then figured it out. But so that's what those are. So if you ever see that, the falx cerebrae, the book mentions dura matter. Dura matter here, okay? Dura, Spanish, English, the same thing. Durable, or it means hard matter or strong matter. And the falx cerebrae, as well as the tentorium. Remember, the tentorium is a tent towards the back of your head that covers the cerebellum. Those are made of dura matter. So when we talk about the falx is in the like this whole section is the fissure, the interhemispheric fissure, and the structure, the connective tissue is dura matter, and when it comes to the middle of the brain, it's called the falx. It's important for us to see the falx for a number of reasons. One, if a falx either in utero or on a newborn, if the falx deviates from being straight, that's probably a mass effect. So then you have to wonder what's causing it to deviate. If it is existent, but there's no brain on either side, that tells you what is going on, as opposed to a skull without brain and without a falx either. That could be a different set of circumstances, a different. I'm running for president of the United States. Can't speak. Anyway, a different disease process. Either one of them, obviously, if you don't have brain, who cares if you have a falx? I don't have a brain. I have a falx, so I'm okay. That's not how it works. Okay, let's keep moving on. So once again, here's our falx cerebrae around here, and then the tentorium is there, except it's more of a tent type structure, which I drew over here before. Okay. Fontanelles, yes, yes, yes. Anterior fontanelle is our #1 location. #2, mastoid fontanelle. Actually, and I think I said before, the posterior fontanelle, you can do the entire exam from here if you have to. And trust me, when you look at some of these babies, the nurses are like, you have to fight with them, and not in a bad way, but it's more of a negotiation of, first of all, this is unusual to see a preemie of this age and a bare hand, and in there, that is unusual to say the least. They are in incubators. For you to get your hand into the incubator, oh my God, you don't want to be my height either. You don't want to be 6 feet tall, putting your hand in an incubator, which was made for a 5 foot tall person. But even then, just putting your hand in the incubator, they're very antsy. They become very antsy because this baby loses so much of a single degree in temperature, and it's dead. So it has to maintain its temperature. It can't be moved. You cannot move the air tube at all. So these are a little scary. But I was thinking about it earlier today, and how difficult it is to see some of the issues that are involved in this exam. And as difficult as the physicality of being in that room with these guys and getting that transducer just right, and sticking your arm into an incubator, and ergonomically it's a nightmare. You don't have to do a lot of these before you become really good, honestly. So these are very difficult to understand on paper, much easier to understand when you're actually doing the job. Now, we mentioned the coronal plane and the sagittal. Sagittal, we're used to sagittal. Coronal. I thought, you know what, let's remind ourselves what coronal means. I have 2 pictures here. The first picture is designed to show you, be careful what you see on the internet. Be careful what you see on the internet. Oh, let me do something here. Okay, so now I can draw. Is this coronal? When you put your transducer, we're doing abdomen right now. When you put your transducer on the Epigastrium. Good God. The epigastrium in this position. Do I ever say coronal? Hell no. That is not coronal. This is not sagittal. This is coronal. This is sagittal. Who the fuck says horizontal? No one says horizontal. That's transverse. All right. And so this is, I was just going to skip this. And I thought, you know what? Let's hold on to it just to show what we're talking about. This one is correct. Where did I steal this from? 1, 2, 3, R, F. Well, okay. By the way, just as an educator, I'm allowed to use copyrighted material any way I pretty much want. Well, within reason, actually. Anyway, that's neither here nor there. Okay, sagittal plane. Yes. Hello, where are you? Okay, sagittal plane. Yes, that is the sagittal plane. If I'm going to do the epigastrium, that's in sagittal. Now, when we're over here on the side of the body, I don't bother. I have never known anyone who calls it coronal liver or coronal kidney. But just so you know, it is coronal. It is technically coronal when you're over here. And when you're transverse, well, transverse is transverse. Transverse there. Even, ooh, transverse. Yeah. Even when you're over here, it's still considered transverse. So transverse, we're correct when we use this term. We are not correct, technically speaking, when we're doing the abdomen. We're way over on the side. We should say coronal, but no one does it, so don't worry about it. All right, why did I bother saying all of this? Because coronal, look at here on the head, coronal is like that. That's how you hold your transducer to get let's just pick this guy. No. That's how you hold your transducer. Let's see. Right, left. This is technically not transverse, it is coronal. On this particular exam, you use the term coronal. You do not use the word transverse. That is just simply the way it is with brain imaging. Okay, let me back up. Let's see where we are at. Okay, I'm going to quickly go over these pictures again, and then as we get into normal variants and then pathologies, we will do a completely different zoom. Oh, looks like I left some stuff behind here. Okay. Cavum septum pellucidum, like I said. Oh, well, actually, no, I was talking about the third. Oh, fuck me. You did not just hear me say that. Yes. Oh, God. I thought I said it wrong. Okay. If you are dead center and you see fluid, it is the cavum septum pellucidum. And over it, this guy here, that is the corpus callosum. This is the bridge between the two hemispheres. Third, and remember, this is not necessarily a part of the ventricular system. Third ventricle is down here. It communicates with I'm just imagining a foramen of Monroe, two of them, one going to the left and one going to the right, lateral ventricles. So that's how the third ventricle communicates with the lateral ventricles. We cannot see the aqueduct of Sylvis, but let's just pretend and draw it in. Aqueduct of Sylvis. Down here we have the fourth ventricle. May the fourth be with you, young Skywalker. And then, oh my God, can we actually see, is this real? I don't know if that's real or not, but that could be the foramen of Uska. Honestly, guys, I don't care, so I don't remember. Don't care about these two. Lushka and then Megendi. I don't care about these. I don't want you to care about them too. I just want you to know that they're there, and that is how things occur. That's how fluid gets around. So we could pretend, let's just pretend and say, yes, that is a tiny piece of the foramen of Lushka, and then the foramen of Megendi is going to come down here and start to fill this space down here, which is the cisterna. Yes, the cisterna magna. Cerebellum. Okay, let's keep moving. Let's keep swimming. Let's keep swimming. So as we've gone, do I have another picture for that? No. As we go off to the side just a little bit, this is a very mature brain. In other words, not a premature baby. It's got plenty of sulci and the other ones. You're losing your mind. Jerry. Yes, Jerry. Anyway, sulci, Jerry. We could see plenty of that. That means this is a mature brain. But we're going to come out a little bit to the side and see, well, a lateral ventricle. The other way we know this is mature brain, this is really thin. Lateral ventricle doesn't get smaller necessarily. The brain around it gets bigger. The echogenic portion inside of this, that's just a little tip of the choroid plexus. I don't know what it is about the choroid plexus. It is like, it's like forgetting the name of the liver. It's not exactly a minor structure, yet you will find next semester that I always search for that name in my brain, and I don't know why. I've got some kind of hang up on it. The choroid plexus, the part that actually creates the cerebral spinal fluid. As we're coming out here, we see the thalamus here in green, which is pretty cool. As we're looking, as a thalamus, oh, you know what? I got a picture of a thalamus in a fetus. Doesn't, when we look at it from a different point of view, the thalamus doesn't look anything like this, but we'll look at that again. Caudate nucleus, boy, that is not exactly a easy structure for us to see. But right here, the caudothalamic groove, this area right here, when we're doing these early brain imaging, this is the most important part of the brain to look at, right there. Trust me, guys, it is easier to do than you would believe. The last time I had to do these in mass, I didn't have a lot of experience, and after the first night of doing 7 of them all night long, literally by the 7th one, I was an expert, and so that's all it took for me to become an expert at it. And that was years ago, and Anyway, as we keep going farther out, we're now into the parietal lobe. I mentioned that we're going to look at the echotexture here in the temporal lobe down there. Okay, so that was the sagittal views. Now we're going to get into the coronal views of the head, the brain. We're almost into the eyes here. So this is way forward in the head, and then the temporal lobe. Well, they're calling them temporal lobes. Yes, I'm not saying they're not temporal lobes, but you go literally 1 more centimeter, and you're going to see basically not the eyeballs themselves, but you will see the bone around there like that. But anyway, sphenoid bone, falx, nice and straight. It's got some growth around the outside, which tells me that this is a fairly mature. And then when I look at this picture, yes, this is a mature baby. This is not a really premature baby, because you can see the gyri going off to the side here. Let's move in on this one. Actually, let's move in on since this one is imaged for us. Let's see, cavum septum pellucidum. Triangle here, we're used to seeing it in OB, we're used to seeing it as a box, believe it or not. Third ventricle down below. Corpus callosum. People, by the way, when people don't have a corpus callosum, this is kind of like if you have a roof on your house. This is a roof right here, this corpus callosum. And when you don't have it, things kind of grow differently, you know. So just like if you didn't have a spleen, your left kidney would grow into that space. Well, if you don't have a corpus callosum, these ventricles can actually grow up into that space and fill that space. Anyway, but lateral ventricles in blue here, caudate nucleus here. I don't expect you guys really to see the caudate nucleus because it is a very small, indistinct structure. We're still going a little farther back in the brain, and let's go to the other side on this one. Here is our echogenic. Wait a minute. I could. Oh my God, I can't remember the word again. Choroid plexus. Choroid plexus, the echogenic choroid plexus. These are filling, these are inside, at least my recent research says that they are actually inside. And certainly, if you're an ultrasound tech of any tenure, you would say that they're inside the lateral ventricles, and the ventricles you're coming down here towards the back of the brain, towards the occipital lobes here. And then as we go even further back, we still have a little bit of lateral ventricle on either side, the falx, and then the tentorium can be seen. I had another picture. I did, yes. Let's look at this. This is in utero, and this is kind of, you get a lot more used to this. Most ultrasound techs get pretty accustomed to this picture. You know, this, I know it doesn't look like a great amount of information, but This is the baby's skull around here. This is the front or anterior, and this is more the posterior. I'll put post here. Let's see, if the baby's lying this way, then, well, you know, right, left. This is actually, yeah, technically, you know, funny thing is you don't know which is right and left because it's a mirror image, and this baby could be aiming in either direction. Now, like the body could be towards you or it could be away from you. You don't know from this picture alone. Anywho, that is neither here nor there. Let's see. Mr. Peanut. Cerebellum. Shaped a bit like a peanut. This baby has an intact vermis. We don't see anything wrong right here. So cerebellum, vermis of the cerebellum. Do you see a worm? Nope, I don't either, but it's there. That the vermis, that structure is really an anatomical structure where you'd have to pull the cerebellum out of the head in order to see the worm-like structure of the vermis. But we know it's there because it's not filled in with fluid. This is the cisterna magna back here. And this looks like a normal size to me. If this person did not have a vermis, we would see fluid come all the way through, because if there's no vermis, it's going to be filled with something, right? There are no voids really in the body. There's always filled with something. So fluid would be filled all the way up to here, all the way up to the fourth ventricle, which, by the way, we don't see in this picture, but still, that's what that would be. Let's see. We're seeing just at this level, you shouldn't see the thalamus. I think we're seeing just a little bit of the thalamus here. Bilobed structure, but like I said, you don't see it clearly because of the exact position we're at, and my wife is vacuuming the floor for the first time in weeks, and she has to do it right now. Hold on. That's okay. Okay. A little comic relief. I'm almost done with this. So, third ventricle on this kid is literally, it's actually not even seen. It is like right there. So that bird creature with the eye thing, it is normally very, 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 very thin. So thin that if we see anything, it is a slight slit in the middle of the thalami, which, like I said, we're not actually seeing the thalami. I think we're seeing maybe just the tiny base of the thalami here. But I picked this picture for another reason. We have a box-shaped structure here. That's actually the cavum septum pellucidum right here. So let's see, back it up. Oh, I've got a couple of, okay, I got a couple of things to pull over. Okay, that is amazing. This, no, that's not it. Okay, let me pull this one over first. Let's get it a little larger. What is this? Oh, this is the cavum septum. No, idiot. Corpus callosum. God, that's once again, I can't stop it, can I? Well, in any case, you can see that is a long structure. There's a structure that if the fibers of the corpus callosum continue on. They don't just stop right there. This structure is not really bounded on the lateral sides like this, because it's a bridge. It connects the two hemispheres, so the fibers continue on. But I guess the structure itself, we call it right here. You can see that it covers over this little tiny. Let's again a little closer. Control. Oh, it won't let me. Well, that stinks. Okay. You can see we're kind of right under there, right under the front part of it. There's a little bit of a space, and that's where the cavum septum pellucidum sits there for a little bit until it's obliterated later in life. That one's not so good. This one is awesome, and this is a video that's a few minutes long and really amazing. So let me tell you about it at first. It's self-reflected. It's basically an art piece, but it is an art piece created by some pretty smart people who put together a model of the brain that is really remarkable to look at. So let's try to run this here. And of course, it wouldn't be a video if I wasn't stopping everything and making sure. Oh, first of all, sorry guys, I just want to make sure that you guys have, I didn't mean that. Okay, I want to make sure you guys have the sound from my computer. So let me open this up again. So let me just, these guys are going to talk about what they're looking at. They're going to talk a little more in detail about what's going on. But look at the structure here of this brain, of our brain. This is really remarkable. Remember when I said that the sulci and the gyri, they create surface area because that's where you do the thinking. And so you could see, this is where your neurons are. This is where you think. By the way, this is the olfactory bulb, which is kind of cool. You actually have like a little bit of brain in your nose, so don't blow it too hard. But this is where you think all around here. And you can see where the surface area, these curves, gives us more thinking area. There is a brain disorder called lysencephaly, where people lack these curves. We lack the sulci and the gyri, and those people are severely retarded because they don't have a whole lot of surface area to do their thinking. And then the entire inside of the brain are just these little highways, these little tracks that run and speak to other parts of the brain, or perhaps go all the way down. If it's a motor neuron, it goes all the way down into the spinal cord, tell you to, well, just scratch your nose. And here, there's a little more. There's some neurons in here, some information. Well, this is the thalamus is in this area, and the caudate nucleus, and all these structures are right here on top of the brain stem. This area bulging out right here is the pons, and the medulla oblongata, I think, is right about there, and that is really primitive stuff. Every animal, every lizard, every dinosaur, every birds, mammals, well, they all have a medulla oblongata. And then, of course, here's our cerebellum back here. Okay, I will play it, and I'll try not to interrupt it again.
All right, so I'm going to give you a little tour of the brain here. So this is it in animation mode. And these are these neural circuitry kind of modules that we've created to show you what the brain might be doing, for example, in the parietal cortex here, as you're reaching out to grab something, where your vision and your sense of space are being integrated. Here we're coming into the somatosensory cortex, where the neurons are communicating to give you a sense of what it feels like to run your hand over your arm, or something like this. Here we're getting into the motor cortex. This is right up at the top of your brain right here. This has some of the largest cells in the body. These layer 5 BET cells, which are sending motor information down through your spinal cord. And we're getting into the frontal eye fields, where you're helping to track your vision to what is relevant in your visual field. And you can see that this is the cortex of the brain. So this is the outer kind of walnut-like covering that's organized into these neural layers, which are sending information up and down. And as we get closer into the frontal cortex, this is the part of the brain which separates us from other primates. This is what makes us human right here. This is the part of the brain which spins on its own. It's communicating without any sensory input, really. I mean, it integrates it, but it's capable. This is what thinking looks like. This is what making decisions and planning your next move and using logic. This is really the most evolutionarily recent part of our brain. And one thing you might notice relative to the occipital, or the rear portions of the brain, so we're right here now, by the way, is that there are a lot more interneurons. The noise of this region is much higher. And what you get when you have a noisier system.
So when he's talking, I know, I had to do it, guys. When he's talking about interneurons, there's a lot more of these side-to-side passageways or communication, a lot more of the side-to-side communication. And that's, those are your memories. Those are how you think and plan. Whereas the other parts of the brain are a little more linear. You have a neuron, and it speaks to maybe a couple of other neurons, and then it continues down its pathway into the rest of your body.
System is more of a potential for optimization. So you can see generally how information travels through the cortex up and down and side to side, and then goes out through these longer range action potentials communicating with other parts of the brain. This guy right here, which you'll see, is the nucleus accumbens, which is where you feel pleasure in the brain. That might be I don't know, eat a delicious ice cream cone or take drugs. That's what's going to be happening there, quite frankly. This is the caudate nucleus, or the basal ganglia. This is where you're integrating all sorts of different things that your brain is processing to decide whether or not you're going to initiate a movement or not. Here's the thalamus, which is the central routing station of the brain. Pretty much everything is going through the thalamus, and it's deciding what is going to be gated and what is going to be allowed to pass to really allow you to bring things into your conscious awareness or not. Down here, we're starting to get into the brainstem. Here's the ventral tegmental area for you brain nerds. This is the pons right here. These are the huge pyramidal tracts which are carrying motor information down from the motor cortex, and this will become the spinal cord when we get down a little bit further. This is the reticular formation here, and these are some nuclei here which are doing things like helping you to breathe, helping you to sense that you're hungry, just really basic reptilian brain kind of stuff, which is controlling basic life functions. Now we're in the cerebellum. This is what's calculating proprioception, where your body is in space and how you can move. This is a particularly elaborate and beautiful part of the brain, in my opinion. In fact, these layers of all these little sparklies comprise half the neurons in the entire brain. There's 50 billion of them. Up here, this is the inferior and the superior colliculus, which sit right about back here, and those are processing hearing and vision, respectively. The optic nerve would be coming in through here into the lateral geniculate nucleus, and these are what are called the optic radiations going into primary visual cortex. This is where you're calculating just super basic aspects of your visual field, like where the angles are, very rough form here, and then that information gets sent up through these wave-like activity into the higher order cortex. So that's what that looks like.
Okay, so I just think that's the most beautiful video, or just not beautiful, but just amazing. And if you ever take a big piece of cauliflower and cut it straight down the middle and split it apart, and you look at it, that's your brain. I don't know why I mentioned that. It's just weird, but so tomorrow, maybe today. Well, next, let's put it that way. Next, we're going to continue on chapter 21. Talk about intracranial. Well, there's some normal variants, and then intracranial pathologies, hemorrhage, which I've already kind of mentioned. Periventricular leukomalacia. Excuse me. Malaysia. That's when you kind of go off to the side and you look at the texture. Agenesis of the corpus callosum, Dandy Walker, I've mentioned. Did I mention Dandy Walker, or just maybe Arnold Chiari? They're all kind of, those are similar. Vein of Galen malformation. Actually, yeah. So actually, then when we get, there's not a lot to talk about, which is good. So we'll continue that next time. That's about it. God, please tell me I am recording. Yes. All right, guys. Until next time. Peace.